Hello, I'm Victoria, and this is my presentation on the technology behind the science of the COVID-19 PCR test. Can you imagine this? It's year 2020, 2021, 2022. COVID-19 pandemic ongoing. You sneeze, get a little cough, and you go off to an urgent care, get a swab up your nose. That sample is sent off to the lab. Bam, science does its thing, and you get an answer on whether or not you are COVID-19 positive. But how does this actually happen? We're going to explore this in this presentation where we figure out the science behind that test that tells you whether or not you have the sniffles or the plague. Today we'll break it down. Here's some key points for my presentation. First of all, we'll talk about what a virus even is. We'll go into details about the COVID-19 virus specifically. We'll go over the timeline for the rollout of PCR testing, how the test works, general lab procedures involved, and interpretation of the test results. Let's dive in. Okay, to start out, let's define what a virus even is. A virus is a microscopic particle, much tinier than a bacteria, which is even smaller than a human cell. Unlike bacteria, which are living organisms, viruses aren't actually technically alive on their own. They, by necessity, need a host because they are unable to reproduce without hijacking the cellular machinery that is capable of replicating DNA. So a virus is actually a clump of genetic material, either DNA or RNA, wrapped inside a protein coat. So a simple way of understanding RNA and DNA is that DNA is like master blueprints for a project, whereas RNA is like a copy of those instructions for a specific task, like building protein. So DNA is master blueprint, RNA project-specific blueprints. And COVID-19 itself is actually a RNA virus for its genetic material. So let's talk specifically about that virus. All right, so COVID-19 is actually caused by a virus called SARS-CoV-2, which stands for Severe Acute Respiratory Syndrome Coronavirus 2. Comes from a family of viruses called coronaviruses, which refers to the crown-like spikes on the surface of the protein. This family of viruses can cause a variety of illnesses, from the common cold to more serious respiratory diseases like pneumonia or SARS. It was first, COVID-19 specifically was identified in late 2019 when the COVID pandemic first started and spread worldwide. So it's primarily spread through respiratory droplets, and its ability to replicate very quickly and its efficient spread through respiratory droplets is what made it so dangerous and efficient in starting the global pandemic that we all experienced. Okay, picture this. It's early 2020. People are panic buying toilet paper, staying home, self-isolating, and it's really hard to know who's sick, right? This is where the PCR test really came in the clutch. It's the unsung hero of those early days of the pandemic. It's able to give patients an answer on, yes, you have the virus, or no, it's just allergies, you're fine, go home. In the early days of the pandemic, scientists and health authorities were able to pretty quickly recognize that accurate testing was crucial in controlling the pandemic and mediating self-isolation when needed. Knowing who was infected helped slow spread because we could have those people quarantined and it helped save lives. But how do we test for something that's so small that it's literally invisible to the naked eye? We utilized a type of testing called RT-qPCR. So this is the type of testing that was already in use. We just had to tailor it specifically to the genetics of the COVID-19 virus. It has been used in research and clinical settings before, but it just needed refinement after we had analyzed the specific RNA of the COVID-19 virus. Within months, RT-qPCR became the gold standard for COVID-19 testing. It's still utilized for rapid testing today. Let's go over PCR testing itself. This is where the magic happens. We'll break it down because I know RT-qPCR is kind of a mouthful, and it's kind of hard to figure out what that alphabet soup even means, right? So let's break it down. RT refers to reverse transcription. This is when the RNA from the virus is turned into DNA because DNA is easier to work with in the lab. This is carried out through an enzyme called reverse transcriptase, which basically generates a DNA script based on the viral RNA that's analyzing. The reverse transcriptase enzyme produces what is called complementary DNA, which is used for the next step, PCR, polymerase chain reaction. So in this step, multiple copies of DNA are made, millions of copies, which makes it easier to detect. So the DNA is amplified using a polymerase chain reaction, which utilizes polymerase enzymes. Makes sense? This technique utilizes primers, which are kind of like sticky ends that attach to specific areas on the COVID-19 virus. These primers you basically act as jump sites for the polymerase enzyme to attach and start copying the DNA. And multiple cycles produce multiple copies of DNA, provided that there was, viral RNA in the sample to begin with. The quantitative part refers to how the test is able to actually quantify the data that's looking at. So it's able to do this by utilizing fluorescent markers, which are added to the DNA as the amount of DNA grows. If it is a sample that contains the virus in the first place, that amount of DNA multiplies with each round of PCR, and the glow gets brighter and brighter. The amount of cDNA is utilized to actually detect whether or not a test is positive or not. So it helps quantify how much virus was in the initial sample to begin with. Okay, so here's a quick overview of how the test works. We get the sample, nurse or whoever swabs your nostril, and then they put it into like this little clear vial, right? That vial contains some reagents which are utilized to bust open those cells to expose any viral RNA that might have been present in that sample. This viral RNA is isolated from any contaminants in the sample, and then some reagents are added to that like tube of your sample. So this includes the reverse transcriptase enzyme, which is utilized for actually producing cDNA from the viral RNA. It's also ran through the PCR, which allows primers specific to the COVID-19 virus to adhere to the complementary DNA. And through multiple rounds of PCR, more and more complementary DNA is multiple copies generated. And then the machine is able to utilize a thermocycler to actually gauge how bright the sample is based on those fluorescent markers, which will light up proportional to how much of the viral genetic material is detected in the test itself. Okay, so let's talk about the test results. So RT-qPCR isn't just a yes or no test, technically. It tells us how much of the virus is present using something called a CT value or cycle threshold. CT value represents the number of cycles it actually takes for the fluorescent signal to reach a detectable threshold where we can actually, as the machine, say, oh, wow, that's a lot. That means it's a positive test. Or, wow, there's nothing there really. I guess it's a negative test. So a low CT value means actually means there's a high viral load because fewer cycles were required to detect the virus. Kind of like a loud shout versus a unclear whisper. Did I really hear that? Is the virus actually there? So low CT value, high viral load. High CT value, less virus, maybe no virus is present, since it took so many cycles to even detect a little bit of RNA. So whenever you hear that your friend tested positive for COVID-19, that basically means that the RT-qPCR detected enough viral RNA and confirmed this through the amount of fluorescence that was detected by the machine. And that's the magical world of RT-qPCR. Remember, stay curious, stay safe, don't forget to wash your hands. Thank you for coming to my TED Talk. And now I should totally know how to turn off this presentation. Play it. All right. Have a good one.